Hi there, we're going to learn about DNA transcription and why it's important to cellular processes. We'll cover the various steps involved in the transcription process, along with where they lead. All known life on Earth stems from the nucleic acid DNA. DNA makes RNA. RNA makes proteins, and proteins make us. But how does the information in DNA get transferred and transformed into RNA? Let's learn. This transformation is called transcription. Transcription is the process by which a DNA segment is copied into an RNA strand, which happens due to an enzyme called RNA polymerase. Importantly, this lets the information of the DNA be copied and sent elsewhere as RNA without causing the DNA itself to deteriorate or requiring the DNA to leave the nucleus. This process happens in three distinct steps: initiation, elongation, and termination. So let's look at these individually, starting with initiation. As you'll likely have gathered from the stage's name, this is the part where the transcription is initiated. In order for this to happen, RNA polymerase must bind to a part of a DNA sequence with at least one transcription factor. That's a protein necessary for the transcription process. The promoter is the length of DNA upstream of a gene where RNA polymerase and transcription factors bind to initiate transcription. And so an RNA polymerase promoter complex is formed here. If you imagine the DNA structure as two halves of a zipper, the RNA polymerase promoter complex is like the fly. Once there's a good bond, RNA polymerase copies the sequence of DNA into an RNA sequence with the help of the enzyme helicase that opens up the wound DNA strands. This phase is sometimes thought of as its own phase, known as promoter escape, but looking closely at this is usually only necessary when there's a problem with this part of the transcription process. So we're left with two single-stranded segments of DNA that are used as templates for transcribing RNA copies. Elongation refers to elongating the single-strand template of our sequence. Because nucleotide bases always pair with a particular counterpart, only half of the DNA sequence needs to be copied in order to be recreated elsewhere. To synthesize RNA, a non-coding strand, or template strand, from one of the DNA halves is used as this template. The coding strand is the one whose base sequence is similar to the RNA, whereas the non-coding strand consists of the anticodons. This template strand gets read by RNA polymerase in the direction of 3' to 5'. However, if the coding strand and new RNA are used as a reference, then transcription occurs from 5' to 3'. And so an RNA molecule is produced from 5' to 3' that is a copy of the coding strand. Termination is how the transcription process ends. This happens in two different ways: row-dependent or row-independent termination. In row-independent termination, the transcription process stops when a new RNA molecule is produced that has a GC-rich hairpin loop. In row-dependent termination, however, the interactions between the mRNA and template will be stabilized by the row, which is a protein transcription factor. Once termination has concluded, the new RNA will get released. Now, you may have heard of reverse transcription. While the name is similar, the process is actually completely different than the one we've just described. In reverse transcription, DNA is created from an RNA template, but this is almost entirely observed in viruses. And crucially, proteins can't ever reverse transcript back into RNA. So the DNA transcription process copies information from the parent strand of DNA into an RNA strand. This strand can then go forth and prosper. This is called translation, and we'll get to that in another video. See you next time.